I'm ready, so start again. And today I'm going to talk about the Schizaeales group of ferns. And this work is actually most part is done by Shosei Owi, the second author. When he was in my laboratory for the master's degree, and now he finished his degrees and working in a publisher. So at this moment, he doesn't do anything about paleobotany, unfortunately. But we are going to make a paper in the future. And also Julian Legrand from Shizuoka University did pollen grains and the spore works. So this is the summarize of our recent advance in this fan group. And holophylogeny is my only my word coined by myself, so no one may know this, and someone may tell that what kind of weird word it is. But I just want to say, as I wrote in the abstract, I want to mean that the fossils are very important to reconstruct Oh,
Well, please could you switch off your microphones, all except of our speaker? Well, and
okay.
by the way, Haru, your presentation is not in a full screen, but I don't know what is this mode, but we can see also miniatures.
Oh, I see. So I will change the style. Just a moment. I just finish once and do it in a full screen mode. I use the presentation mode. So where is it?
Don't worry, we will wait. Oh, now it's okay.
Is it okay?
Yeah.
Okay.
Yeah.
Yes. So using this word, I want to express my thought that the fossils are very important to reconstruct the true phylogeny of the plants. So I don't know whether this word holophylogeny is exactly good or not. I'm not an English speaker. So anyway, I just start using this word today. And this is the example of our material, permineralized materials in calcified concretions or sometimes in silicified material. In this presentation, I'm going to introduce you 5 new specimens from Japan, southern Chile, and also Antarctica. And also, finally, I will show you the preliminary result of our phylogenetic analysis using the molecular phylogeny and the fossil data. And the left is the example of the late Cretaceous segments containing sporangia and spores. And the right one is from Japan, early Cretaceous material. And first, I'd like to explain briefly about the living groups of Schizaeales. There are 3 families recognized, and in the broader sense, Schizaeaceae is sometimes used in terms of Schizaeales. But this time, I'm going to use Schizaeaceae in a strict sense in the middle. And the particular feature of the Schizaeales is that they do not have typical sorus. Otherwise, each sporangium bone on the dorsal, ventral side of the segment solitary, and usually making 2 lines along the midbrain. And in lygodiaceae, only in lygodiaceae, there is indusium for each sporangium. And the whole Schizaeales have sporangium with terminal complete annulus. And the shape of the sporangium is somewhat pear-shaped, but the orientation of the sporangium is different in each each family. For example, in the lygodiaceae, the sporangium is lateral with a short stalk, and the Schizaeaceae and Emeritus, they stand up. To make anatomical comparison or morphological comparison, we have to determine the structure, fatal structure in morphological term. And here we use solophore for the set of sporangia lined in 2 lines on a very narrow structure segment. And the length of the segment is different in species or sometimes in family. And in the lygodiaceae, usually, as you know, they form elongate fertile structure. And here we call solophore of like this structure. And in other family, for example, Emeritus, the Emeritus forms a typical fertile lateral pinnae, usually standing upright. And each pinnae have this kind of segments, and each segment is like a short solophore forming 2 lines of sporangia along the midbrain. So someone says the earlier workers use different names for these structures, sometimes solophore and sometimes ultimate segment, ultimate fertile segment, etc. And we are going to compare these later. And this is the Schizaeaceae, and they form elongated structure. Here we call this structure also solophore. The last one, this long one. And the many different terms are adopted to the same fertile structures. There's a long history, and sometimes solophore or sometimes sporangiophore or both adopted by some authors. And I don't have much time to explain briefly, explain well about this, so I'm going to skip this table, and then I'm going to talk about the recent phylogeny of Schizaeaceae. These are based on the molecular phylogeny, basically, and this is a time tree. So in the evolution of ferns, particularly the leptosprangiate ferns, in which Schizaeales are included, the leptosprangiates probably started from Carboniferous, and also eusprangiates a little bit earlier. And this figure is used to explain Japanese Triassic Narua flora. So here's a line of the Triassic area flora, but this flora, this figure is just I use to show the phylogeny of vascular plants. So just to forget this Nariwa flora. And this triangle shows the fossil evidence, start of the fossil evidence of angiosperms. And the Schizaeales are one of the two major groups of leptospongiate ferns, and the one group contains Glyciniales, Dicteriales, Matoniales, or Hymenophyllaceae. And the other group, Schizaeales, are rather primitive group, and then divided Hydropteridalis and Schizaeales tree ferns, and other mostly recent ferns. Within the Schizaeales, the relationship within the family or between the genera are well resolved now by using molecular data. And it is typical that there are three groups consisting of one with LIGO-DEC group, and the second one here, Schizaeace group, and Anemoaceae group. So the three families, LIGO-DEC, Schizaeace, and Anemoaceae, are well defined using the molecular data as well as the morphological data, as I explained earlier. But these are the molecular phylogeny, basically, and we do not know anything about the early diversification of Schizaeales ferns. And when we talk about the origin of the Schizaeales, we have to go back to the Carboniferous leptospongiates. And the morphology of these Carboniferous ferns are quite different from the present ones. And we paleobotanists cannot explain exactly between the relationship between the Carboniferous types of ferns and the later Mesozoic types or recent ferns. So there's still deep morphological and morphological gaps for our understandings of the relationships of the Paleozoic and Mesozoic or recent ferns. So this is the example of the Carboniferous ferns, Semptembergia, famous groups, and which are compared to the Schizaeales. And this, because the sporangia are pear-shaped with terminal annuals like this. So I do not mention much about this, about the Carboniferous ferns. And this is just a table that show you how many numbers of fossil morphologies are compared in this study. 49 or so. And this is the list of the new specimen, which is in the abstract also. But in the abstract, I didn't put another specimen from US formation, ALY23, which will appear later in this presentation. And the Wakayama, in the middle south of Japan, there is a early Cretaceous strata bearing some familiarized plants, concretions. And this is the Barremian material, Barremian to Botulinum, Barremian to Aptium in sometimes. And another specimen is from Hokkaido, Japan. Japan is very, Hokkaido is very well known since the first work by Mary Stopes and Fujii in 1910. And since then, there are many reports about the plants preserved in the concretions, calcareous concretions. And the new specimen is in one of these concretions from Mikasa Formation, and the age is Coniacean Santonium. And another one is from Antarctica. And I made an Antarctic survey in 2011, and I found one silicified material in the Livingston Island, and I will show you today. These are probably Point Williams from Point Williams Formation, Cinnamonian to early Campanian. And other two specimens are from south middle of Chile, near Concepcion, and the fossil site is named Cocholge. And here there's a Maastrichtian marine sediments, like Hokkaido shallow marine sediments, and producing the calcite concretions from Cranilaue Formation. And we found two different specimens of probably each represent different genus. And at the final part of this talk, I will show you the preliminary phylogenetic analysis, result of phylogenetic genetic analysis. And we used 19 different characters based mainly on the sorophore or fertile part structures, anatomies, and sporangium anatomy, and also spore anatomies, spore morphologies. And here is the first example of the already described permineralized fossils we know. And this is Schizaeales by Stops and Fujii. And this photograph here is a new material, not the one I will show as a new material today. This is the specimen secondary found by Yoshida and described by Yoshida and Nishida before, and which is identified also as Schizaeales mesozoica, the same species that Stops and Fujii described. And this one has a beautifully preserved fertile part, and the leaf segments enclose the sporangia. And the reconstruction is probably like this. This is the fertile structure of line drawings of the anemia, a recent species, but the structure of Schizaeales is quite similar to these structures. In this illustration, you cannot see typical leaf part lamina here. Lamina is reduced in this illustration, but usually the anemia has segments with flat lamina and with dorsal, with ventral sporangia. And this is a living one, which I found in Mexico. And Schizaeales has spores of Schizaeales spores, and with fine muri in the distal face, like this. And this is another permineralized specimen, Paralygodium. This is the first one is described from Hokkaido also by Yoshida and ourselves in 1997. And this one is a reconstruction, suggested reconstruction of the fertile part. And this paralygodium has a palmate segmentation of front or fertile part. And it is each segment recurves to protect the sporangia like this. And the spores are 40 micrometers or so, and the surface is rather flat without very fine typical structure. And interesting thing is that paralygodium is later found in the early Cretaceous of Canada and also from the Eocene by Stocky and Carafit. So this fossil genus has a rather long history from the Cretaceous to the early Paleogene. These are the comparisons of the different species of paralygodium. But I don't mention deeply about this today. And from here, I'm going to show you 5 different specimens, new specimens in our laboratory. The one is Stachypteris-like fertile structure, which we call solophore here. And it has, these are serial sections A and B, and you can see the middle vein here. And in the other section, you will see the typical complete annulus on the top of the sporangium. And the segment shows typical solophore structure. And the one peculiar feature of this specimen is the spore morphology. And this one is very close to the Ischiosporites type spores, and which is found in Stachypteris type structures. So based on these morphologies, we tentatively identify these as Stachypteris-like solophore. But the Stachypteris is known from the compression impression fossils mainly, and probably this is the first example of the permineralized, anatomically preserved fertile structure of Stachypteris groups. And later we found another specimen from Arida nodules. And this is the cross-section of the solophore. And here's a midrib, and you can see the segments, lamina, narrow lamina here. And the sporangia type is the same as the one I showed earlier, I showed before. So Stachypteris type fertile structure. And the second one is another Schizaeales from Hokkaido. This one is similar to the Schizaeales mesozoica that Stops and Fuji and we found earlier. But the difference is that this one doesn't have a coiled structure. I mean, the segment is flat, and also the each sporangium has terminal apical capsules like this in Schizaeales. And the number of these terminal cells are important feature to identify species. So this one has a larger number of apical cap cells in compared to Sphaeropteris, Schizaeales Mesozoica. And the spore morphology is also different. And there are muri on the distal side, but you can see the reticulate structure between the muri. So based on these structures, we decided to describe this as a new species of Schizaeales. And the third one is from Antarctica. And about this specimen, I made a presentation in Brazil, IOPC, in 2016 in Brazil. And this one is from the Williams Point of Isla Livingston Island here in the near the Antarctic Peninsula. I went there thanks to the help by Chilean Antarctic Survey. And the Antarctic Survey of the Chilean base is next to the Russian base. So I also went to see the Russian colleagues there. And in the Livingston Island, along this coast, we walked along, and near these seals, I found a satisfied specimen, which are not in situ rock, but the material is from early Cretaceous probably. And this is the nodule satisfied rock I found. And immediately when I found this, I noticed this one. And these are the remains of fragments of fern sporangia and fertile structure. So I took this and made sections. And this is the example of the sections of the similar types of rocks. I found actually 3 different rocks at that time, no more yet. But one of these structures, I made peel sections, and these are photographs of peel sections. And as you see, the preservation is very fine. And these are vertical roots in the soil. And these are the sections of vertical roots in the soil. And you can see tiny fragmentary structure here. These are root hairs of these roots. So the preservation shows root hairs and even the mycorrhizal structure attached to the root in the different sections. And the material is probably embedded in the volcanic ash or volcanic sediments and silicified. And from that fertile structure that I found on the surface of the rock, I made serial sections of peels first. And taking photograph of these peel sections, we made the reconstruction, morphological reconstruction using Amira software. And I will show the movie of the how to make the Amira reconstruction. Oh, sorry. There should be just a moment. I have to come back to the Oh, yes. Here you can see the reconstruction of the few sections. So the result is shown on the left side. So we made these reconstructions like this. And so this is the suggested reconstruction illustration of the fertile structure. And you can see the sulfur structures, which is coiled to the inside. And we also use the different method to reconstruct or to make the images of the rest of the specimen that I found. Because I made serial sections of nearly 100 slides, and there are some other parts left in the rock. So for this structure, we used Malpis. This is a new method that we call Malpis. The name is shown on the right side. I cannot see the letters because of the control panel of the presentation. So you can see, you can read probably. And this method is designed by Professor Yasuhiro Iba, Dr. Yasuhiro Iba of Hokkaido University. And we have been collaborating with using this structure to see the inside of the rocks. And Malpis is a quite simple way. Just cut the rock and then start to grind the section surface automatically in the thickness or interval more than 0.1 micrometers. So depending on the thickness of the grinding. And we continue these grindings many times. But each time we take very high resolution photographs and then combine these images and reconstruct the inside. Then we can choose different materials preserved in the rock. For example, ammonites and other things. So everything in the rock we can see by using this technique. Because peel method only you can see the material that are not destroyed by acid. But this one, you can see the whole structure of the rock. And this is the example of the Rhinichat material that we are now doing. So if you see the particular specimen in the rock, you can extract it digitally and make reconstructions. And this is the example of the Malpis image from Antarctica material. And you can see the section of wood. This is probably some kind of seed. And here there is a fertile structure of the fan that I showed before. So first, in the larger magnification, I will show you how the images are obtained. So this is a very short movie, but we can continue if you continue grinding as much as you like. I will show you again. So these woods are probably the wood of some gymnosperm conifer root wood. And these tiny structures are the lateral roots. And sometimes these tiny grains are probably the mycorrhizal structure, fungal structure. And this is the Marpes image of the close-up of the fertile fern structure. So you can see the stipe here and branching segments and each sporangium preserved. So the image resolution is very high. So if you find the spores inside the sporangium, you can see the details of spores also. I do not see each spores this time, but you can see it. And about this Antarctic material, we made a preliminary taxonomical comparison, and these are the summary of the structures. And each sporangium has 70 to 25 cells of the annulus, and spores are preserved sometimes. And the preservation is not so good always, but the tetrahedral spores have a rather smooth surface. And the terminal capsules are also shown. And this one, sporangium shape is lateral and similar to Lygodium. And the spores are also similar to the Lygodium type, but the Lygodium has indusium for each sporangium, as I showed. But this fern doesn't have indusium. So this one is typically different from Lygodium, but similar to Lygodium or Lygodiaceae. These are the spore morphologies, and these are the examples of living Schizaeales. The Lygodium has this kind, and Moria and anemia has different types of spores. And the anemia spores are quite similar to the Schizaeopteris spores. And the Schizaea has a bilateral spores, as you know. So this one is similar to, most comparable to Lygodiaceae, but X-induced. So probably the different genus of these groups are close to Lygodiaceae. So about the phylogenetic position of these fossils, I mean the Antarctic material and the Paralygodium and the Schizaeopteris. We put these in these places preliminarily. And this one is a kind of illustration that Gary Rothwell calls the Christmas tree. So you have the phylogenetic tree of the living species, and this one is this phylogeny is provided from Wickstrom and others in 2002 paper. So they compared some fossil specimens in their studies. And for example, Lygodium beerholstiana or Stachypteris that I showed the fertile structure like this in the tree. But these are not based on the detailed comparison. And even our color dot, the three different groups of ferns are put on somewhat closest place in this phylogeny. So this is not the holophylogeny, just a Christmas tree. And the final result of our analysis will be shown at the last slide. So before that, I will show you two other specimens from southern Chile. Here's a Concepcion, and Chile is a long country like this. And the Cocholgue is located a little bit north of the Concepcion. And we continued the excavation since 2007 several times, and 2011 and 2010, big tsunami wave hit here also, and these houses are almost destroyed, and the basement raised to 1 or 2 meters, and the Cretaceous Kirikina Formation is distributed along the coastline, so we can see the elevated base of the Cretaceous sediments later than 2010. And on the Cretaceous bed, there's a Eocene bed, which contain coals and silicified peat. And in the silicified peat, there are well-preserved abundant plant specimens also. But I do not show it today. These are Maastrichtian, and the conclusions are similar to the Hokkaido material, the shallow marine sediments, and contains ammonites. And this one elongated ammonite, Bacillus type of ammonites, and the black or brown ones are plants. So one of the example of the material from this conclusion, I only show you only one example. This one is a series of sporangium, but each sporangium is covered with a leaf segments. And I put this image upside down, actually, because this one is compared to the living groups of Lycopodium hupertia, which is growing pendant. So this is the part of the this fertile spike. And actually, this is the upside down, or morphologically, this one is the right structure, right orientation, and the microphyll is here, and sporangium and spores inside. So there's no living groups of hupertia in Chile anymore. So this is the example of the warm period of Chile in the Cretaceous time. And from the same conclusion, we found two different kinds of structures. So the one is this one, and the reconstruction is somewhat preliminary, but you can see the sorophore structures. And the interesting thing is that this specimen has indusium, and the structure of the spores and the sporangium are similar to Lycopodium. Lygodium. So this one is comparable to LIGO-DAC. And this one is quite similar to the previous one, but this one does not have inducium. So there are different kinds of Schizaeales plants in the Cretaceous of Chile at the end of the Cretaceous. And the spore morphologies and the sporangium morphologies are different from the previous ones also. And for this material, I used the amina reconstruction. But this one is very preliminary, and the movement of the reconstruction is somewhat it's not good yet, but I will show you. Yeah, so this is the preliminary one, and you can only see the mummy-like structure. And using these 5 different new materials from the world, we used the we made the phylogenetic analysis using the computer software that I showed in the abstract, the typical TMT method, and the molecular data is used for the constraining. So this is a last slide showing our present result of the reconstruction. And the carboniferous materials are not compared to this analysis because they are quite different from the other Schizaeales. So the analysis is, as I see, well resolved the relationship of the living and fossil specimens. And importantly, the 3 different families are well resolved in this analysis. And the Moriaceae, Anemiaceae group are here, and the Schizaeaceae is here. And there's no good fossil material that can be compared with the Schizaeaceae. And the LIGO-DAC are here. And the many new material are located in the, in my word, good position in this phylogenetic tree. And the Schizaeopteris, which is compared to the Anemiaceae, is located here. And the Schizaeopteris mesozoica, the first one, and the new species is in the sister relationship. And the Chilean material is combined together, but as I see, as I said, one of the Chilean material, SR1, has inducium, but the other one doesn't. So anyway, these are close to the Antarctica material. And Paraligodum meckeliti, this is from Canada, and other Paraligodum are grouped with these fossil groups. And the Lygodium is made a good monophyletic groups, and this one is typically characterized by the presence of inducium. So in our analysis, we used the solophore for these all fertile structures. And the inducium evolved only in lygodium, but in cilia material, one is X-induciate and one is with inducium. So sometimes these evolved in parallel. And the solophore structure is not found in Moria, but this one, Moria, is probably interpreted as the reduced types of solophore structure. So this is the all of my talk today. And although we will do something more in detail, probably this is the best result we have by now.
Thank you. Спасибо. Thank you, Haru, for your brilliant presentation. So many novelties, technically and in scientific content. And now time for questions. If anybody wants to pose a question, raise your hand, your own hand or your virtual hand. Switch on your microphone and please ask. Well, my first question, just of curiosity, how it was in Antarctica? Simply cold or impossibly cold?
Yeah. So, of course, it was in summertime there. And because there's no Japanese bases in the Antarctic Peninsula, I asked the Chilean Antarctic Survey to take me to the Antarctica. And the director of the Antarctic Survey of Chile was my good friend. And finally, he invited me to come. And also in 2016, I also visited Antarctica again. And the typical sediment you cannot see because of the glacier and ice. But the specimens probably came from the Cretaceous sediments. So we have many things to do in Antarctica, but it depends on the vice versa things, because if we want to dig more fossils, global warming is a problem. So I don't know the future, but at least we found a very good sample in Livingstone Island.
We have two comments in the chat from John Marshall. Many thanks, interesting techniques, good to go, and just like this from Joe-san to everybody.
So I talked about the Malpis method twice, 2 or 3 times in the international meetings, just briefly. And the machine is typically, there's only one machine in the world by now. So now it's still working. And we started grinding the Reinhardt material. And now we are getting some good results. And one of the results we presented in the last IOPC in Czech. So I'm sorry that you are not coming. So
yes, nothing to do. I hope for better future, for
yes,
times.
I hope. Yeah, actually.
As to this technique, you do sections, not virtual sections, but just normal sections.
Just to make a section or you can start grinding from the surface of the rock anywhere. And the size is also, you can even grind the rock about 15 or 20 centimeters big. So, and it goes totally automatic. So it may take several days or a week to grind certain thickness. But
Is it possible to make it virtually via tomography or something, or the material does not fit to this technique?
The problem is that the material disappears because we grind everything. But inside is we can get very high resolution image, and you can pick up each plant material and reconstruct it. And the tomographic material is quite different because they do not destroy the material. So the problem of this method is that if you find a very tiny material, everything disappears. So I started discussing about the typification of this material. So we can, for example, in the line chart example, you can make a 3D reconstruction model using the image data, but the real fossil is not preserved. But fortunately, in line material or some of the Hokkaido material also, the specimens, there are many specimens. So you can choose one for grinding and others for typification sometimes. But we have to discuss very deeply about this in the future.
Well, I see two questions in the chat. Could you also look in them? Evgeny Karasov asked you, could you clarify the name of the software? Amira or Amira?
This one is quite different. That one, for the serial sections of the pills, the Amira. The Amira is used for many different materials, and typically it is used in the medical sciences.
Zhenya, have you satisfied the answer?
Yeah.
Amira.
Yeah.
This one, yeah.
Amira
So this is a widely distributed software. The problem is that it's a little bit expensive for the paleobotanists.
This is not free software, it's paid, yeah? Premium software.
Not free, expensive.
No, very expensive.
Yeah,
this is commercial based.
I know the free open source alternative of the similar software.
Oh yes. Yeah. So if you can use this free software, it is okay also.
One more question from Hanzang Song. Thank you, Professor Nishida. Is this method using laser cutting or a purely mechanical method? Will it destroy the tiny structure of the fossil?
Yes, totally mechanical. So I cannot see you how the machine works, but the machine, you set up the rock vertically on the machine, and then there is a diamond plate to grind the surface, and then you grind the surface with a lot of water also, and then take photograph and grind and take photograph. So quite technical way.
Well, I do not see all of you, and so if anybody want to ask question, just make it loudly because I fear I can miss somebody. Well, we have one more thanks in the chat, and I have one more my own question related to spores. To photos of spores, I've seen somewhere in the middle of the presentation there were black and white photos, and I wonder how did you do them? Yeah, this
one.
For example, this one. Is it a scanning electron microscope?
No, this is a laser image.
Aha.
Laser micro.
So you made it without extraction of the spores from the.
No, you cannot see. You can take photograph in the slide.
Cool.
Yeah, in the peer material, you can do it. And this one is done by Julien Legrand. Julien, can you explain something? Are you here?
Yes. So, yeah, these two pictures were taken with a confocal laser microscope. And so you don't have to make any special operation to take the grain out of the slide, but you just can use your slide made for for example, phenological preparation or for the pin method, and observe directly on the microscope. So that's very convenient, in particular when you have some very rare grains that you want to have the more detailed picture. So you don't need to make some picking of the grain.
Thank you.
Yeah. So
Thank you.
these photographs are also the laser image, yeah?
Yeah, this one.
Yeah. Yes. But the first, this one is scanning electron microscopy.
Well, I do not see if anybody else wants to ask something to our speaker. So, last chance. Thank for your interesting talk. It's Evgeny Karasov writing in the chat. Well,
okay.
if
I have some.
Yeah.
I have some chat letters, and can you send these chats to me later?
Женя, мы можем послать это позже? Чат к Шхару. Just a moment.
Yeah, of course.
Yeah, we will do it.
Yeah, because I have to answer to the Van Koninberg work.
Okay. Thank you once again, Haru, for your interesting talk.
Thank you for all of you.
Thank you very much. It was very interesting.
Thank you.
And I see more thanks in the chat.
Oh, yeah.
Thank you.
Okay, so
Thank you for coming and see you next time.
Yes, спасибо again.